Hi everyone, thanks for joining our talk today. My name is Rochelle Belanger and I'm in the biology department at the University of Detroit Mercy, and I'll be co-presenting this research talk with my undergraduate research students Karen Crile and Sara Abdulelah. We're going to talk to you today about some past and current ongoing work in our research lab, which seeks to examine the effects of atrazine on the chemosensory system and looks at chemosensory deficits, DNA damage, and changes in cell morphology after environmentally relevant exposures. We use crayfish as a model organism to examine these changes post-exposure. So atrazine is an herbicide that's been used in the United States since 1958. It's an agrochemical which is heavily applied, especially in the US Midwest. And you can see this map over on the right-hand side from the USGS, which shows the estimated use from agricultural land in the United States. And we can see a really heavy band of atrazine use in the Corn Belt region of the US Midwest. And it's heavily applied to corn, sorghum, and sugarcane crops to increase yields by controlling broadleaf weeds. Approximately 80 million pounds of atrazine is applied annually in the US. It can, however, enter into local streams and rivers through runoff, seepage, and regional transport, and it's been shown to have effect on aquatic organisms that are exposed to atrazine in these habitats. Now, fish have been shown to have decreased in food odor detection as well as grouping behavior, and it's also been shown that we can have an increase in bioconcentration of atrazine in these aquatic organisms that are found to have exposures to atrazine in the environment. Although the US EPA has set an environmental limit of 10 parts per billion atrazine, we can see here from this figure that atrazine has been recorded in the environment well above 300 parts per billion, and this elevated concentration of atrazine in local streams and rivers can last for as long as 3 weeks, leading to increased exposure concentrations and length of time for organisms found in that environment. In our research lab at the University of Detroit Mercy, we study crayfish and the effects of atrazine on crayfish. And we use crayfish as a model organism for studying the effects of atrazine on aquatic organisms because crayfish are polytrophic. They are a keystone species, which means that they transfer energy from the benthic environment to the pelagic environment, and they play a pivotal role here in the aquatic food chain and the transfer of energy. So any changes in their population size can have far-reaching effects on the food web. They rely heavily on chemoreception for survival. They're active at night. They're often found in murky waters, and therefore visual signals are decreased in these areas. So they rely on chemoreception for finding food, avoiding predators, finding mates, and for finding shelter. So any impact on their chemosensory abilities can affect then population size and therefore other species as well. In order to investigate how atrazine affects chemoreception in crayfish, we collected crayfish from the field, we brought them back to the lab, we allowed them to acclimate, and then we exposed them to environmentally realistic concentrations of atrazine for 96 hours. We then transferred them to a Y maze like you see on the bottom right, and we investigated time spent at food and mate odor sources. Using an overhead camera, we also looked at locomotion and time spent in each arm of the Y maze. We then transferred crayfish to clean water for 72 hours and recorded responses every 24 hours during that 72-hour period.
So, following the methods described by Dr. Belanger, the results were illustrated in the bar graphs to the right. In the top graph, we can see that the atrazine-treated crayfish spent about half of the time at the odor site compared to the control crayfish. On the bottom graph, we see that even when recovering from the 96-hour treatments, the experimental crayfish still spent less time at the sites than control. These results showed us that the environmentally relevant exposure to atrazine, which was 80 parts per billion, had an effect on the chemosensory abilities of crayfish with regards to food and mates. The results also told us that even following the exposure, crayfish could not fully recover their chemosensory abilities even after as long as 72 hours. So now, following the previous results, our group wanted to dig deeper into the olfactory organs to see how olfaction was affected following atrazine exposure. We hypothesized that since chemosensory responses were diminished after exposure to atrazine, this would mean that the olfactory sensory neurons could also be damaged. So our methods consisted of collecting the crayfish and treating them in separate groups for 7 days. So we did a group of 3 to 4 animals exposed to 10, 40, 80, 100, and 300 parts per billion, and of course our negative control of 0. Following treatments, we collected the crayfish's weights, euthanized them, and dissected their olfactory organs, which were the lateral antennules that are shown in Figure A. These antennules were then fixed in paraformaldehyde, decalcified, and cryosectioned in a sagittal manner. So in other words, they were sectioned from top to bottom so that we can see as much of the structures shown in Figure B, including the olfactory sensory neurons. Then, following the sections, we performed a tunnel assay, which is an experiment that will help us indicate DNA-damaged cells. So these cells would be called tunnel-positive cells and will fluoresce green. And with our hypothesis of more DNA damage in a dose-dependent manner, we also predicted more green labeling. And as you can see, during the tunnel assay, we added DNAs to the positive control shown to the right, and it resulted in lots of green labeling. So we predicted that our atrazine-treated crayfish, especially the ones that are 100 and 300 parts per billion groups, would have the same type of fluorescence. And in fact, the results proved our hypothesis. So you can see that in the negative control, no tunnel positive cells are detected. And as we gradually increase the level of atrazine, we also gradually increase tunnel positive cells, which are indicated by the white arrows. The blue labeling is our counterstain, DAPI, and this stains the cell bodies. So in other words, this stains the total amount of cells available in the tissue, while the tunnel stains the DNA damaged cells. So with these stainings, this led us to our next experiment with the tunnel images. And this was to manually count the damaged cells in order to determine the percentages to compare the damage of different atrazine levels. So basically, we counted tunnel positive cells and divided it by the amount of DAPI cells. And as a result, we see that there are 0% tunnel positive cells in our negative control as predicted, then an almost linear increase in percentages of the tunnel positive cells in a dose-dependent manner. And this experiment further supported our hypothesis that atrazine causes DNA damage in cells of the lateral antennules, including the olfactory sensory neurons.
In addition to studying how atrazine affects olfaction, we also wanted to study how atrazine affects the hepatopancreas of the crayfish. The hepatopancreas, shown in the bottom image, plays a key role in detoxifying toxins like atrazine out of the body. Because of this, studying the hepatopancreas is important since damage to the hepatopancreas can impair its ability to detoxify in the future. To study this, we treated our crayfish using the same methods used in our olfaction study. We treated crayfish with 10, 40, 80, 100, and 300 ppb atrazine for 7 days, as well as performing a 0 ppb control during the treatment period. After treatment, we rinsed, dissected, and fixed the hepatopancreas for further analysis. We performed a standard hematoxylin and eosin staining in order to examine vacuolization and tubular epithelial damage in atrazine-exposed crayfish, and furthermore, we also performed a tunnel assay in order to investigate whether hepatocytes in the hepatopancreas were undergoing DNA damage following atrazine exposure. Images from our H&E staining did show that the hepatopancreas had morphological changes following atrazine exposure. As atrazine concentrations increased, we saw an increase in vacuolization per lobule, and this is indicated by the asterisk shown in our images here. We also noted an increase in disintegration of tubular epithelium with an increase in atrazine, as well as pronounced nuclei of lobular cells too. Preliminary results from our tunnel assay did show that atrazine does cause variable amounts of DNA damage to the hepatopancreas following atrazine exposure. When looking at our 10, 40, and 80 ppb tissue, we do see an increase in tunnel positive cells as atrazine concentration increases. However, when looking at the 100 and 300 ppb tissue, we noted little to no tunnel positive cells here. We are not quite sure as to why this is the case, but this is something that we do want to investigate in the future. Taken together, the studies done here in our lab have shown that atrazine ultimately has a negative impact on crayfish. Earlier studies in our lab have shown that crayfish do lose their chemosensory abilities following atrazine exposure, and additionally, crayfish are unable to regain their chemosensory abilities 72 hours post-atrazine exposure too. We also saw that olfactory sensory neurons in the crayfish experience DNA damage following atrazine exposure, which does align with our behavioral studies done here in our lab. Lastly, our current work in our lab, focusing on how atrazine affects the hepatopancreas, do suggest that atrazine does negatively affect the hepatopancreas. Knowing this, having impairments in chemosensory abilities as well as the detoxifying processes done by the hepatopancreas can negatively impact crayfish's growth and survival. Moving forward, we want to continue our work focusing on how the hepatopancreas is affected by atrazine exposure. We would like to perform a time and concentration series to help determine a dose response. We'd also like to examine caspase-3 expression, as caspase-3 is a protein that's upregulated during apoptosis in order to determine whether tunnel-positive cells are undergoing apoptosis too. Lastly, we want to determine whether cells in the hepatopancreas can regenerate following atrazine exposure. So with that, we would like to thank current and former researchers that have worked here in the Belanger lab, as well as acknowledge the Barry Goldwater Scholarship awarded to our lab member, Sara Abdulelah, the University of Detroit Mercy, and the Rebuild program funded by the NIH for funding the studies mentioned here in our talk, as well as thank Dr. Paul Moore from Bowling Green State University for providing help with the studies done here. Thank you.